Hello, my name is Adam Moule from the University of California, Davis, and today I'm going to be talking about a rapid and diffraction limited photolithography of organic semiconductors. Our main problem that we're trying to solve here is that organic semiconductors to be used in any sort of a device would be need to put into a pattern. And currently there isn't a technique available that allows us to pattern on a sufficiently small length scale, meaning less than a micrometer. So if we were to talk about a toolbox that we would need in order to have a good patterning, we would need to be able to layer OSC films from similar solvents, and we developed that a few years back. We need to be able to etch OSC films with light without damaging the polymer, which we developed a couple years back. We need to control the locations of dopants using light, another thing that we've developed a couple of years back. Um, we need to be able to use all of these steps in an easy, fast, compatible roll-to-roll coating. All of the steps need to be diffraction limited in resolution. So in this case, it would be smaller than the wavelength of light or in the order of hundreds of nanometers. And finally, our process would need to work for all semiconducting polymers, not just one semiconducting polymer. So we developed a series of steps that allowed this to do these steps in one or two polymers. And in this talk, I want to talk about how we're going to generalize it and get to this point where we can really use our system on all semiconducting polymers. So this is the setup that we were going to be talking about for the entire talk. We have a glass slide. We've deposited a semiconducting polymer film onto this slide. We have a solvent layer on top of the film. The solvent layer is generally about 100 micrometers thick. And then finally, we have a cover slip, and the whole sample is transparent. We illuminate the film with a laser through the top, and this laser is focused on the film surface. And the observation that we have is that the film dissolves where the laser touches up into the solvent, and then so we're left over with a pattern wherever the laser happens to touch the film. So now the first thing we need to get to is what is the mechanism of this particular effect? And the first mechanism that's really important is this light-induced dedoping reaction. So what we have here is that we have, for example, the polymer P3HT, the dopant F4TCNQ, and this doped polymer is always in equilibrium with the condition that we'd have an undoped polymer and an undoped dopant. It turns out that if we shine 405 nanometer light onto a sample in which this equilibrium exists, that light can be absorbed by the neutral dopant, and in the presence of THF results in a reaction that gives us this molecule. This molecule is not a dopant for P3HT, and so essentially what shining a light on the sample does is it dedopes the sample. And so in that case, we'd be using a nonpolar solvent. And so this salt structure right here is going to be non-soluble in a nonpolar solvent. If we destroy the dopant and make this molecule over here, the dopant is removed from the film, goes up into the solvent. We're left over with a neutral polymer, and that neutral polymer will dissolve in a nonpolar solvent. We thought this was the whole mechanism. This was like the whole deal up until about a year and a half ago. And about a year and a half ago, we started doing some experiments in collaboration with Professor Grigopoulos from University of California in Berkeley. And he has an instrument that allows them to use two different colors of light. So they would have one color of light in which they're exciting the sample at high energy, and then a different color of light in which they're probing the shape of the feature that you made with the first light beam. And then this allows us to do an in situ measure of the etch rate as a function of the position. So this is our dissolution time versus the power. And what we show is that we have a discontinuity. So basically our dissolution time is going down as we're increasing the power. And then there's sort of a step function improvement in the dissolution time, followed by, again, a steady reduction in dissolution time with increased power. And so this step function here, that was really strange. If we're only working with a single dissolution mechanism, there should be a smooth curve. And we have a clearly non-smooth curve here, and it makes us think that maybe there's more than one mechanism at play. So we studied this, and the way that we studied it was by using two different excitation lasers. If we used a 400 nanometer excitation laser or 405 nanometer excitation laser, the light would be absorbed by the F4 TCNQ, and so we would be electrochemically or photochemically dedoping the sample. In the case of the green laser, in this case, the green laser is not absorbed by the F4 TCNQ at all. It's only absorbed by the polymer, and so the only effect of the green laser would be heating. So now, if we look at writing with a 405 nanometer laser and the 543 green laser, we can see that we actually get a pattern in both cases. And what that means is that we're able to pattern the film using the temperature difference, the temperature rise that we get in the film because of the green laser. So we studied this for a number of different intensities for the two different reactions, and we modeled the reaction rates. And what we found was that something like 95% of the dissolution with a 405 nanometer excitation is due to this photochemical dedoping. On the other hand, 100% of the dissolution for the green laser is due to a temperature change, or this dissolution because of the F4 TCNQ being heated and then diffusing out of the film. And so we have two different mechanisms at play, just depending on how we excite the sample. So now we would like to be able to try to quantify what our dissolution rate is. So we're looking at dissolution rate as a function of the excitation laser, as a function of the intensity of the excitation laser, and as a function of the dwell time of the excitation laser. In each case, we're going to scribe a line in the film by dragging the laser over the film. The polymer will dissolve into the solvent, and then we use an AFM to look at the line shape. So here is a series of cross sections for different line shapes for this 37 microwatt and this 133 microwatt excitations as a function of the dwell time. And this is for the this is both for the 405 and the 543 nanometer excitation. And what we're going to do is we're going to take this data and we're going to plot the line width. So that's this dimension right here at half height. Oh, sorry, the line width is a function of the depth. So basically at 0 depth, we have a thick line, and at full depth, we have a thin line, and that's changing for each one of these. So if we plot that, what you can see is that for each of the plots of the data at the different dwell times, we're getting a flat dissolution rate. And so if we then fit this dissolution rate to a kinetic model, so one kinetic model would be proportional to the intensity of light, another kinetic model would be proportional to the intensity of light squared. What you see is that slope is proportional to the intensity of light squared, or what it tells us is that our etching rate goes up as a function of the square of the light intensity. So now the other thing that we can look at is as we look at absorbance through the film, it's clear that both the liquid P3HT and the solid P3HT will absorb light at 405 nanometers. However, if we're looking at absorbance through the solvent, now there's a solvochromic shift in the P3HT such that the liquid state P3HT, the dissolved P3HT, doesn't absorb light at 543 nanometers. And so again, we have a difference in the two wavelengths and how we would model this data. In the case of the 405 nanometer excitation, we know that the solution, that the P3HT in solution is going to be still absorbing 405 nanometer light, which means it still heats up. So we can develop a finite difference model where we're paying attention to the shape of the laser profile, the intensity of the laser profile, turns out the broadening of the laser profile. We can then model the temperature rise of the sample as a function of radial distance from the center of the laser profile. And then given our knowledge of the activation energy for dissolution, we can calculate out the dissolution rate. And so what's really interesting is you can see that the temperature rise spreads out pretty far across the sample, but the dissolution rate is really, really localized to the place where the beam interacted with the sample, because we have a very nonlinear dependence of dissolution rate on the temperature. If we then simulate and measure the line profiles for this absorbing solution case, what you see is that our simulations give these very, very narrow lines. If we compare it against data, you can see that the data gives a much wider profile. Again, this is only about 500 nanometers wide, but it's wider than we would expect. What this tells us is that we could still get higher resolution than we've demonstrated if our laser was better focused. In the case of the green light, in this case, we have the non-absorbing solution. So the solution doesn't heat up with more light exposure. This leads to an interesting problem, and that is when we first turn the laser on, it heats up very much at the center of the laser spot. But as the film starts to dissolve, it gets thinner. And as the film gets thinner, it absorbs much less light. And as it absorbs less light, it heats up less. And so we wind up getting a self-limited resolution for the non-absorbing case, essentially because it doesn't get hot enough at the edge of the laser profile in order to continue dissolution. And again, this is because it gets thinner during the dissolution process. All right, so I hope I've convinced you that we can figure out how well that's working and that we have a rate law for our process. Now I'd like to show you what we can do with it. And I'm going to give you the simplest circuit demonstration I can imagine, which is a nanowire. So we coat our polymer, we coat the dopant with the orthogonal solvent, we put it into one of these solvent cells with the methyl THF, we illuminate with a laser, and we illuminate in a pattern such that we're leaving a wire between two gold electrodes, but then getting rid of all of the rest of the material that might connect those electrodes so we can get direct measurements of conductivity along the wire. We made about 10 wires this way. This is one of them. This is a laser scanning confocal microscope image. And what you can see here is a nano wire that goes between the two electrodes. This particular nanowire is about 250 nanometers wide, about 40 nanometers thick, and about 40 micrometers long. We can then take these nanowires, like I said, they're doped in solution, but we have an electrochemical method or a chemical method for de-doping, and we can completely de-dope the polymer. And then again, we could expose that wire to a dopant again, and so we can recover the high conductivity that we get from doping. What's important to show here is this is a fully reversible process. We can remove the dopants, we can add the dopants, we don't lose any of the material. Finally, this lithographic method enables us to pattern in any form factor. So when I'm talking about wires, you can imagine a circuit that has a wire with an L bend. You can imagine a circuit with a T in the wire. And what I'm showing here is that not only can we make wires, but we can make wires in any shape we like by removing the rest of the material. I'm also showing cross sections for these wires. So there's 1, 2, 3, 4 positions. And what you can see is that these wires are quite narrow. Their width is on the order of about 200 nanometers, so that's less than diffraction limited. This wire here is kind of flat on the top. This one here is not so flat on the top. But again, at this resolution, the difference in the width of these two wires essentially comes from the drift of the laser inside of the laser confocal microscope, or in other words, our pattern is as fine as the instrument will allow, and if we had a better instrument, we could write a better pattern. So with that, I'd like to thank my funding sources. This was originally funded from the Department of Energy and then has been funded for about the last 5 years through a couple of NSF grants. The critical scientists that developed this were Ian Jacobs and June Lee from my first batch of students, and then more recently, Zaira, Tucker, and Goktuk have been working on it. And with that, I'd like to thank you for your attention. And of course, I'm asking you for questions, but what I really mean is I'll answer them at the question session.